Welcome to Natural Habitat Adventures Daily Dose of Nature. Today's presentation, Secrets of the Ocean's Sponges, is brought to you by NatHab Expedition Leader Melissa Silva. Melissa is a marine biologist who has been guiding nature adventures in Mexico since 2012 and recently joined NatHab's team in the Central Highlands to lead monarch butterfly migration expeditions. She focused part of her studies on nudibranchs and is currently working on her master's thesis on seahorse mating behavior. Based in Puerto Vallarta, Melissa combines her marine biology expertise with broader naturalist knowledge to share wildlife conservation through nature encounters with travelers from around the world. Melissa, thank you so much for being here with us today. It's your show. Take it away.
Thank you, Rob. Thank you for the introduction. Well, thank you everyone for being here. Let's start with this. As Rob already mentioned, I'm a marine biologist, and well, this is something I really like to talk about marine organisms, in this case, the sponges. And I was just telling Rob that I think this is a very nerd webinar, so I hope you stay with me all the time and hope not to lose you in any of the information I give you there. But as he says, we have the question area, so you can write any questions you have regarding the topic. When you think about sponges, when you are in your home or in your city and you think, I need a sponge, usually this is what we think, right? Usually is what we are used to think of when they say sponges. The dish sponges, the sponge to wash our car softly and polish it. The facial sponges, those are like very important to clean our faces. Even for some artistic moments, for my kids, for example, I used to have a lot of sponges to paint or to have shapes on the sponges. And I think one of the most popular sponges in the last decade is SpongeBob SquarePants. I'm pretty sure you have seen or heard about this TV show, the cartoon show. And I want to open a parenthesis here, a quick note about the show. The creator of that show, the producer and creator, used to be he was a marine biologist. So I don't remember the name right now, but if you make a quick search, you will see that all of the characters on this show represent a true animal, a real animal from the marine environment. So it's a silly show, but I like it. Okay, so I close the parentheses and we go into what brought us here. When I think about sponges, usually together with the sponges I have at home, I think about the sea sponges. And on the previous webinars I was working with, if you join me, or you can check them later, I spoke about the reefs, I spoke about corals and other sessile benthic animals that are in the ocean. And one of the most important through the readings were the sea sponges. They can create reef, they can be the foundation of reef ecosystems, later on colonized by corals or shared with corals and algae and other organisms. The sponges that live in the ocean, there are more or less 9,000 different species described to the date. There are some that are extinct. So if we count the extinct ones plus the ones still living nowadays, there are more than 10,000 different species of sea sponges. And pretty much all of them, 99% of them, live in seawater or brackish water. Very few sponges, like 150 species maximum, live in fresh water only, like lakes or streams or ponds in freshwater areas. So well, when we say about sponges, we usually refer to sea sponges. So the sponges, as they are sessile, they don't move. They were thought to be as the plants until very recently. They said, like, well, it's another type of algae. It's a weird plant of the ocean. Until 1765, that Mr. John Ellis he was an Irish naturalist, and he was very passionate about like describing species, and he actually had a lot of letters with Carl Linné, the father of taxonomy, to put it on a way, the one that uh, started the nomenclature for all life beings. So with this exchange of letters and information, he described the well, corals and the seaweed and sponges from the Irish and the British coasts. Uh, he described how they move the water through their bodies, something that of course plants don't do. You don't see them moving the water through their bodies, pretty much like breathing as an animal does. But it was until almost 100 years after, in 1833, when they actually gave the name or the status of animals and a specific phylum to the sponges. So this sir, he was a sir from the London University, I think, Sir Robert Grant. He was an anatomist and zoologist, and he described like almost on microscopic detail the structures of sponges, and he gave the name of porifera to the sponges because porifera just translates completely to they have pores. So this is something that I find to be very funny, because usually in science, if you study the nomenclature, the taxonomy, and of course common names of animals and plants, they are like very simple. In Galapagos, for example, and in Puerto Vallarta, we have the blue-footed booby, the bird that has blue feet. So it is so funny just to see like, okay, they have a lot of pores in the case of sponges, and Sir Robert Grant named them porifera because they have pores. They are thought to be the first multicellular animals. I will go deep into why they are animals, why they are multicellular, why we think they are the first. But there are some studies, here we have two of them, that found some biomarkers, some chemics that are called steroids 20-C-methylstigmastane, that is present in a certain group of marine sponges. So they found this steroid biomarker in rocks that date back to 600 million years ago. So that's before fish, that's before fish, before sharks, before dinosaurs. I mean, like way, way, way back in time. To illustrate this a little bit more, I have this semicircle with the life, the explosion of life, the evolution of life on Earth. The very first, let's say, living organisms were prokaryotes. Prokaryotes are pretty much cells without nuclei, without organelles inside the body, inside the cell. So all the usually is RNA instead of DNA. All of these compounds is mixed inside the cytoplasm, inside the organism, the little cell body. Through the millions of years, they started evolving, they became photosynthetic, they became a little bit more complex, and then they decided to encapsulate the different parts of their internal body of the cell. And is when we have the eukaryote cellular organisms. At the very beginning, we had, I don't know if you heard of an organism named cyanobacteria, and archaeas, and the protozoans. So all of those organisms, they are microbes, they are microscopic, but they have like complex cells, not to put them as true cells, but just to say they have compartments within the cell that have different jobs. See, so those are the eukaryotes. So the eukaryotes, they have nucleus, they have the ribosome, they have the Golgi apparatum, and eventually they just started coming together and making clumps and becoming more complex, and some cells get specialized, and is when we have the multicellular organisms. We have multicellular algae, multicellular fungi, and multicellular animals as the uh, sponges. Okay, so well, I hope I had some clear explanation on this. I'll go back later for that. So sponges are very simple animals. They don't have tissues, they don't have organs, but they have eukaryotic cells. So that's why we have them within the animal kingdom. Okay, their body plan is very simple. You can see this is like a longitudinal cut of a simple sponge. They have many holes, a lot of holes, the pores around the body that, well, more technically, they are called ostia if it's a lot, if it's a plural, or ostium if it's a singular. And they should have at least one osculum. One osculum is one big pore through where to which the water goes throughout, with wastes and with CO2. Remember, they are animals, so they do breathe. They take oxygen from the water and they expel or exhale carbon dioxide to the water. Okay, so we have the pores, the holes on this porifera, ostia and osculum. It can be one osculum, or or they can have many oscula. The water will go in through the pores, through the ostia. It will mix in the atrium. It's like a bigger compartment through in which we have like all the water that is going out, and then through the osculum. And the nutrients that they catch, they can go in the mesohyl. The mesohyl is like a sandwich layer of collagen and like a gelatin-like substance. Okay, we have here a zoom in of the mesohyl. In this moment, just imagine we are going very tiny little. We are microscopes, and we will see what's going on inside the body of the sponge in a cellular level. Okay, so what we see here is just a little tiny part of a sponge body wall. This is the mesohyl. There are a lot of specialized semi-independent cells because they can move. Some of them can move through the mesohyl in different areas, and some of them are totipotents. They can metamorphose, or they can change to be others. I will go pretty much into each one of the cells, because this is why we consider sponges animals, because all of this complexity. Okay, one of the most important, if not the most important, maybe, cell that sponges have are the choanocytes. The choanocytes are also called collar cells because, well, once again, they have a collar-like structure in one of their areas. And these choanocytes are fixed in the inside of the sponge. So we have an external area on the mesohyl and an internal area. The internal area is the one that gets all the water and moves the water to filter it. Here you can see in this little diagram, the food particles get attached, or they get trapped in the mucus of the collar, and then they are phagocyted into the body to other cells. Once again, remember, they don't have organs, so they don't have stomachs, they don't have intestine, they don't have digestive system at all, respiratory system, nothing like that. So everything is going to be through either osmosis, that is just passing one substance or one particle to another side through the cellular membrane, or like in this case, phagocytosis, that they actually get the food particles embedded or surrounded by the cytoplasm of the cell and absorbed and eventually digested inside the cells. Okay. And this is a beautiful, I just love this photography, micro photography of different conocyte chambers. So these conocyte chambers, you can see here on the left side of the slide, CC, marked as CC. They are all over the place, and there are a lot of canals that bring the water in or out to the different chambers. Here's a little bit more of a close-up of the conocyte chamber, and you will see each one of those marble-like or grape-like the structures. Those are the conocytes, okay? And you will see all of the flagella in this area. Something very cool about the conocytes is that they can transform into spermatocytes. So they can modify their structure to be mobile and free, and then eventually they can be released in the water, look for another sponge of the same species, and fertilize the eggs. I'll go there in a moment in reproduction strategies. Some other specialized cells that the sponges have are the pinacocytes and the porocytes. These are pretty much like external cells or structures, because pretty much the pinacocytes are in the outside part of the sponges, and the porocytes are some specific tubular cells that are, let's say, in touch with both areas, the outside and the inside of a sponge. As the description here says, the porocyte is the cell in charge of closing or opening the ostia, the little pores, so you can have water coming in or nothing coming in, or going outside, or through going in the cavity, in the sponge cavity, or or protecting the body from pollution or from chemics from other competitor to put it on away. This is another micrography. I really love it because there are other type of pinacocytes that are on the inside of some sponges. So it depends like where they are. We can call them exopinacocytes or endopinacocytes. Inside, endo, exo, outside. And this is a beautiful description or illustration of a pinacocyte cell. The cell body is right here, so the nucleus will be here, and the cell makes this structure like a shield type of structure on the outside of the sponge. This is another type of photography, microphotography. Here is like a diagram of what I'm talking about, like the pinacocyte and then the other formation, the body, and the porocyte. Here on the photos here, what we are seeing is the porocyte from above. I don't know if you can notice that. So you will see this dotted line is the ostia, the ostium on this porocyte. So what we see is the porocyte from above, and all of the cilia that will help the porocyte to open and to close the pores. Other type of cells are the amoebocytes, and as the name says, amoebocytes looks like amoebas, like these protist organisms. One theory that a lot of researchers have, a lot of scientists set up in a lot of these articles that I'm showing you and others that I read, is that the sponges are considered the first animals because they took different organisms that were already out there in the ocean, and they just were able to organize and rearrange these organisms that were living freely into specific cells for a greater body. So the amoebocytes are thought to be amoeba-like microbes, super old, very old, that were phagocyted, that they were absorbed by a primitive mesohyl, by a primitive sponge, and they turn out to help this very new organism. So the amoebocytes, they are like messengers. They can take food, nutrients, they can take information from one side to another on the mesohyl. But the coolest thing that they can do is that they can differentiate into any other of the other cells, any other type of the other cells. Archeocytes, sclerocytes, spongocytes, colonocytes, lymphocytes, at least, those are all the types of cells that the amoebocytes can turn into. See, so they have this capacity. If you want to put it in a more common way, they are stem cells of sponges, amoebocytes. And here I wanted to show you why we call the sponges to the sponges. They are made, some groups of them are made of a protein very similar to the collagen that is called spongin. And you can see all of these little structures here. All of that is what gives the sponge its fluffy, spongy texture and appearance. Okay, and the spongiocells the spongocytes or spongocells in a moment of their life, they were amoebocytes. Another very important and specialized cell is the sclerocyte. Some sponges are squishy and some others are a little bit hard, and some are like super hard because they have these spicules that the true name are spicules, that's the true name. I was thinking of thorns on the plants. The spicules of marine sponges are made either out of calcium carbonate or silica. There are some sponges that their body has more calcium carbonate and they are extremely hard. They pretty much are calcareous organisms, like shell, pretty much, because it's the same material. The spicules can have like any different shape. Let me show you that era this one. All the different shapes that we have on spicules, and as I mentioned, they can be made out of silicon or calcium carbonate. With the spicules, the people that knows, researchers, they can tell you what species of sponge we have on the sample. So they do know if this belongs to a extinct the freshwater sponge from Northern Europe, and if they see this other one, they will tell you, no, this is a species that we still have nowadays, but it's endemic to this island or this or that. So the spicules are so specific that they can determine species on the sponges. And once again, we go back to the cells. There are some specific cells that are the responsible of making the spicules. We have pretty much like two types, the founder cells and the thickener cells. So here is pretty much like a diagram of how they will work. So one of them will start putting all together either the silica or the calcium carbonate, and the other one will try to make it bigger or longer or wider. So hence the name thickener or founder cells. See? And then we get to the oocytes or the egg cells or reproductive cells. As I mentioned, the choanocytes will become eventually spermatocytes or spermatophores. But the egg cells are specialized cells that are only, let's say, feminine reproductive cells. See? Most of the sponges are hermaphrodites. That means that they can have both reproductive cells within them. Remember, they don't have gonads neither. Gonads are organs, and the sponges don't have organs. So they do have cells that are specialized in the reproduction. What happens when it's time to reproduce? When we need more baby sponges in the ocean, part of a sponge or a whole sponge decides to modified its guanocytes, so that sponges or that single sponge will expel the sperm to the water, hoping to get to a female sponge, to a receptive sponge that will absorb, here you can see the diagram, will absorb the sperm cell in the mesohyl and will place it with the oocyte, and of course with fertilization, division, etc. And eventually we will have, or well, the sponges will have a mobile larva. The mobile larvae, this is something very, very cool, because hopefully you have seen a human larva in the very early stages of development. It looks just like a human larva to me. When right before we are becoming the blastocytes, it's just like a bunch of cells, a morula, and it's just the same thing. So all of these tips or these key moments on the development of an organism is what help the researcher to tell if one thing is an animal, a fungi, or a plant. So we have in common the development of the larvae on our reproduction. And well, the larvae on sponges is mobile. They move. They are free swimmers, and eventually, through the time, they will settle down to the bottom and well, become an adult sponge. This is a beautiful photo that exemplifies what I'm telling you. It's a giant barrel sponge. This is from the Great Barrier Reef in Australia. And this is something very cool. It's not only there, but the photo is taken from that place. In the Mesoamerican Reef here in the Caribbean, we have also this kind of sponges, this species. And what I know is that when they have the spawning moment, it coincidentally happens with other species spawning, releasing eggs and sperm to the water. And that is when we have the whale shark coming to feed on this area. So it's linked, the spawning of sponges, fish, and other organisms on the Mesoamerican Reef is linked with the arrival of whale sharks. So another thing that why they are important to the ecosystem. The asexual reproduction of sponges can be in three different ways: budding, fragmentation, or gemmulas. The budding and fragmentation pretty much are similar. The budding is that pretty much like they start growing a baby from their body, and then that baby separates from the parent or the original sponge, and it's pretty much a clone of that sponge. The fragmentation would be that if a fragment of the sponge is taken, or the sponge breaks or whatever, if they have amebocytes, if they have these totipotent cells that can transform into others, if they have those amebocytes on the fragment, they can regenerate and create a huge new sponge. But this is pretty much just cloning, because they have the same DNA and genetic information. The gemmules, on the other hand, are a little bit more sophisticated than fragmentation and budding, because they really need to create a new body. I don't want to say organ because it's not an organ, but it's a gemmule. It's the spores some fungi make to reproduce. All those spores, this is pretty much the same thing. They have totipotent the cells, the amebocytes, or here archaeocytes. They are covered in a membrane with spicules that protects them, and usually they have the gemmules when there are some abiotic factors that are stressing the sponges. You can call it a descent or an increase on the temperature of the water, is the lack of nutrients, more competitors, more predators. So what they do is they create these gemmules. The gemmules are free, and they can be dormant for a while on the substrate because these are not free swimming particles or larvae, and they will be on the substrate, and eventually when the temperature and the abiotic factors are just right for them, they will start developing into new sponges. Okay. Bueno. There are other cells. I didn't want to show many of those, but one that I thought was very important are the cyanobacteria. So they do have, like other living organisms, the cyanobacteria, there is a complex whole organisms itself, living inside the mesohyl, living inside the sponge body. They have many other jobs there, or work doing. Most of them is for photosynthesis. Some of them have the job of protecting the sponge from diseases. They have fungical, antibacterial, and antivirus properties, these symbionts that live inside the mesohyl. Bueno, let's go back. We go big again. We leave our microscope in a side. Remember, this is like the general body structure of a sponge, but actually they are more complex than that. There are different body plans for sponges. Some are more complex than others. The researchers say, scientists say that the asconoid body plan would be like the most primitive and simplest of them all. And from there we go with more complexity and with newer species of sponges in terms of evolution. So, well, the ascon or asconoid organization, now you know what are the pinacoderm, the pinacocytes, the choanocytes. So it's the simplest one. They just have the chamber, the atrium, where all the water goes in and then goes out through the choanocytes and out to the water. Okay, very simple forms. Usually these are small sponges. They don't tend to be very, very big. And usually they have a calcareous spicule, or a calcareous, more than silica, they have calcium carbonate in their spicules because they need more protection. Since they are so simple, they tend to be thin. Then the syconoids or sycon body plan, they get a little bit more complex. If you notice, they have canals inside the mesohyl, and those canals are flagellated. So pretty much outside they look like larger asconoids, but they have thicker body walls because of these complex invaginations, they call it. So you can see it's pretty similar to the others, but they tend to be a little bit bigger and of course thicker in their body wall. And then one of the most complex is the leuconoid or leucon body plan. And this is series or flagellated chambers that are interconnected with little canals, one to another. So with this type of sponges, with this type of body plan, the sponges can grow more, like in complexity, but in size also. Like this sponge species. And from this type of sponges, from the leuconoids, are the sponges that we associate mostly with the human use. I will go in a moment to that. Okay, now, why sponges are animals? If we are not convinced on why they are animals on a cellular level, let me tell you a little bit about the phylogenetic explanation of this. So we are all alive. We are all part of the life. We are all part of the kingdom Animalia because we have the complex cells. We are eukaryotics, and we have different cells doing different stuff within our bodies. And that's it. That's the only thing we have in common with the sponges. All of these phylums or phyla that we have, cnidarians, echinoderms, arthropods, mollusks, and chordatas, that it's us and everyone with neural cords. I'll go there deeper there if you want, but all of the chordatas, echinodermatas, mollusk, we all, and even the jellyfish, the cnidarians, we all belong to a bigger group of animals named Eumetazoa or Eumetazoans. That means that we do have organs, we do have more complex systems and organization in our body, and porifera don't have that. So it's a very thin area in which we can tell porifera, the sponges, are animals. They are animals. Okay, very quick. We'll go over the taxonomy of porifera. There are four extant four classes. There are two that are extinct, and there are some species over there that are like dancing without a specific class because we just have very tiny fossils or pieces of their spicules or their bodies, and we are still, well, scientists are still working on that. From the Demospongia class, pretty much more than 70%, almost 80% of the species of sponges belong to the Demospongia, and pretty much all of them are leuconoids. They have this complex system of chambers, very diverse. They live in every level of the floor in the ocean, and most of them have a silica based spicules, so they are more soft rather than hard as the calcareans. The calcareans, as I mentioned, they have the spicules made out of calcium carbonate. They can present any distribution in their body plans, asconauts, leuconoids, and these have a very specific or a very important role in ecosystems because they can form reefs. So if we have a group of sponges that are calcareans, they will create a solid foundation or solid base for other cnidarians or for corals, polyps, to settle there and start growing and create even more room for more species. The Exactinellida class is a group of sponges that pretty much live in deep sea only. They are very beautiful. I've seen some of the photos. I invite you to look for this, and they are very popular. I just read that for as decor on weddings. So I don't know if the photos I saw were like real sponges from the Exactinellida, or if they were like fake sponges emulating this type of glass sponges. On this specific group, I found this very interesting information. I invite you to look for the article. It's online and free access, and it has a interactive video. But they were studying the disposition of how they establish the sponge establish the spicules, the internal skeleton, or let's call it skeleton, but remember, it's not a true skeleton, but specialized structures. So they were checking that this type of structure, the disposition, tends to be more resistant to compression and to weight. So they can resist, I don't know, like 2 or 3 times more compression than other type of organization, and also like 2 or 3 times longer structures with a lot of strength. So check on that one because, well, we still are working on that. A lot of people still studying the sponges because of that. And then we have the class Homoscleromorpha. This used to be inside the Demospongia class, but well, there are some variations, And now they know by molecular analysis that they are completely a different class. And this specific class, it's being studied to know how the epidermis can regenerate. In general terms, regeneration, but more than anything on epidermal tissue, the pinacocytes. Bueno, so if all the information I gave you is not enough to love the sponges, let me tell you why they are important, why we should appreciate them even more than now. They are home for many species in the ocean. I want to say that word, but I cannot pronounce. Myriad, millions of species. So fish find a perfect, soft, and protective bed to sleep, to lay eggs, to mate. Here on the photo that you can see on the left corner, the bottom corner of the slide, the white stuff, the white flagella or threads that you see around the sponge, actually those are worms. So some type of worms that they create this calcareous protection, they can go around the sponges and they have this substrate that allows them to get more nutrients and a better position on the reef. This other right bottom photo on the slide is one of the glass sponges that I was telling you about, the wedding decor. And they use this specific, the name is Venus flower sponge or something like that. It's Euplectella spergillum. And the thing, it's actually this one. Oh, see, I do have it. Is this one. It's the same sponge, the Venus flower. And what happens here is that a couple of these shrimps, deep water shrimps, they go inside when they are larvae, when they are still very tiny. They go inside the sponge. They start growing because they have food there, they have protection, and there's a moment in which they cannot go outside. So to mate, they have to call the opposite sex, a smaller one. The partner goes in there, the partners grows, they are mating inside, and there is a moment in which they are too big to go out of the sponge, and they live all of their lives inside that single sponge. So this is like a very romantic way of seeing that until death do us part. So pretty much that's why they use it for weddings. Then some other sponges are used as hats, like this, as a shield. The Dromidae family of crabs use these sponges that are, remember, they have the spicules. So spicules are, they harm the mouth of any animal that might want to eat or take a bunch of these. So the crab knows that and takes the sponge as a camouflage, of course, but also as a protector, as a protection to against the predators, against the bites of predators. And just for you to notice, this is a female with a bunch of baby crabs, a lot of eggs between her legs. And then one specific, I'm pretty sure you are familiar with this, the hawksbill sea turtle. This is an specialized, when they are young, they can feed on algae, sponges, is the cnidarians, seagrass even. But when they are fully mature, when they are grown-ups, they exclusively feed on sponges on the reefs. So the Redmochelis imbricata, the hawksbill sea turtle, it's something that the biologists call ecosystem engineer, because with the help of the turtle, the sponge is, well, they feed on it, is destroyed, but other sponges can grow from either the same species, from the same sponge that is being eaten, or from other species that were the competitors of this specific sponge the turtle is feeding off. So they form the reef, they give more room to other organisms or even to the same species of the sponge. And well, this is another thing that is super cool because the dolphins in Shark Bay of Australia, in Australia, they learn, or they, well, yes, they learn to use a protection when they are fishing, when they are hunting the fish, or crabs, for example, that they tend to bury and use stuff on top of them to protect against predators. So the dolphins, is a Turpsius species, is like the one of the common dolphins, bottlenose dolphins could be, or a relative to those. They hold, or they place their beak, their upper maxilla, in the sponge, in a very soft sponging sponge, and with that they go to the bottom, they dwell on the bottom, and they can catch stuff without harming their beaks. So here you can see there's a lot of rocks, a lot of broken shells that are very sharp. So that's what they do. And what the researchers of these studies learn is that adaptation that the dolphins have, it passes from generation to generation, from mothers to daughters. And then, well, of course, the males on the group, they will learn to do so. But this is something so cool, that not only humans use sponges. And well, we have a lot of biomedical compounds that we take from sponges. Most of them have anti-tumoral, like antiviral, and anti-cancer drugs specifically. Here we have some of the compounds. I didn't want to go deeper on that because biomedical topics are quite a thing, very complex. And well, some of the species that we can have, that we know that we can get the biomedical compounds from there. But not only were used like in this like molecular level, we use them also as a whole. Since ancient times, since the Greek, and before the Greeks, since Egyptians, and I'm pretty sure before that, without a historical record, the sponges have been used for cleaning, for hygiene, and to soften, for example, during the wars I'm just thinking about Alexander the Great. They used to have this type of sponges on the saddles of the horses, in between their helmets and their protection to go to the war, so they don't have the blisters or extra protection against, I don't know, arrows or whatever they were fighting with. Menstrual sponges are a thing. We are still having like a boom in menstrual sponges. I strongly recommend you to go and do a research, because there are a lot of cons and a lot of pros. So take an informed decision on that. A very specific use of the sponges that I find to be not very nice to know is that the ancient Romans used to have something called tersorium, that is nothing but a sponge tied to a stick, and they use it to clean their butts. So instead of toilet paper, they have the tersorium, and it was a public tersorium. So I don't go any deeper. I won't go any deeper on that. You can read the rest and imagine the rest. And well, nowadays, of course, bath sponges are very popular. They are very soft. They are hypoallergenic. They can hold a lot of water. And there's a huge industry of like harvesting, but on their sustainable practices. So if you're going to buy one of those, just check that they come from a sustainable farm. There are some in Florida, some in Greece. And I wanted to make a special mention of these that are not real sponges, because while I was doing the research on that, in Spanish, I look for esponjas, and I had these two examples, the zacate and the lufa. Both of these are vegetables. They are like true plants, land plants. The zacate comes from the agave fibers, and it's just the fibers, you can see there, like twisted and compact all together. And the lufa is actually a cucurbite. It's first cousin of cucumbers, to put it on a way. So it's a plant. It's completely a plant. Inside the loofah that we use, there are the seeds. I don't know if you have the chance to see them like a little bit fresher, So they have black seeds inside. That's why we know these are not real sponges. And I think that's it. And we still have some time for questions, Rob. Thank you, everyone.
All right. Thank you, Melissa. So before we dive into the question and answer session, I'd like to remind everyone that if you do have questions, just go ahead to the question field in your control panel. If you go to the upper right-hand corner, click on those three lines, it'll open up the menu and click on questions. So let's get to these questions. Can you tell us why these sponges are so brightly colored sometimes? How does that happen?
The coloration, it can be just pretty much like their pinacocytes, their skin cells that have pigments in it. Remember I mentioned the symbiotic organisms that are inside, like cyanobacteria and those. So here, for example, the cyanobacteria, they can have greenish, bluish coloration, but if they absorb other or other microorganism in a symbiotic relation, they can express the color of those microorganisms. So pretty much is because of that. So that's why, for example, the deep water sponges tend to be more dull in their coloration, or white, completely white, like they don't have any need. Some of the researchers say that the colors they express is pretty much like our skin, that they protect themselves from the UV light, from the UV rays. So
Oh, that's very interesting. So you showed a slide explaining emerging life forms, and the description mentioned something about sponges and fungi. Are fungi a part of the plant world? Or are they related to sponges in any way? Can you clear that up a little for us?
Well, they are fungi. They have their own phyla, their own kingdom. I mean, we have kingdom, just to put it like in a very simple way, kingdom Animalia, kingdom Planta, which is we have vegetals and all the green stuff we see. We have animals, plants, we have bacteria or archaea, like all the ancient microbes, and we have the fungi kingdom. So they are so special. I think it's one of the most marvelous organisms that we have in the world, because they are not plants, they are no animals, but they do behave and they do have characteristics of both of those. And even the microfungi, the micro organisms on the fungi kingdom, they look like either one or the other, like a plant or animal, but they are neither. So yeah, they are in this specific kingdom of them.
Great. Thank you for clarifying it. That it's just a fascinating subject.
Yes.
So tell me, Melissa, what's up next for you? What adventure is your next adventure?
Well, we are getting ready to go to the kingdom of the monarchs. So I'm been diving in the kingdom of the ocean right now. A lot of webinars about the sea world, but we are getting ready. The season starts in very soon in December. We have the monarchs arriving hopefully for November the 1st. I have some contacts in the US and Canada, and they told me that they already saw a bunch of them flying south. So that's what I'm into right now. I have still some webinars coming, one each month. If you want to join me, please. Thank you. And yeah, pretty much that's what's on near future for me.
Wow. Well, we look forward to seeing you again. Melissa, thank you so much for this incredibly interesting presentation.
Thank you.
And I'd like to thank everyone who joined us today. So if you are interested in information on how you can travel with NatHab and even head out to Monarchs to see Melissa, please give us a call at 800-543-8917, or you can send us an email at info@nathab.com. Our adventure specialists are happy to help you out. Join us tomorrow for our next daily dose of nature. You can check out this week's lineup, including registration links, on our website at nathab.com/webinars. We did record today's presentation, and we will have the replay available on the website soon. With that, I will conclude today's webinar. So long, everybody. We will see you next time.